Hello, this is Josh Patel back again with another biology video. Today we're going to be finishing up chapter 8, which is from DNA to proteins. And we're going to start with 8.5, which is translation. So before this, in the previous video, we went from one copy of DNA to two in replication. And then after that, we went from DNA to mRNA, which was transcription. And now we're going from mRNA to proteins or polypeptides in translation. So our key concept is translation converts an mRNA message into a polypeptide or protein. Amino acids are coded by mRNA-based sequences. So amino acids help us form proteins, and we get those by the codes in mRNA. So translation converts mRNA messages into polypeptides, and a codon is a sequence of 3 nucleotides that codes for an amino acid. So here we have our segment of mRNA. This is just a little image of an RNA. There's way more of these segments. So a codon is a set of 3 nucleotides, so 1, 2, 3. Right here is a codon. Oops. AUG is a codon, and it codes for MET, which is a polypeptide, and each 3 codes for a different thing. So this is how we get our proteins from. The genetic code matches each codon to its amino acid or function, and there's one start codon, which is that methionine, and there are 3 stop codons. So we don't have to memorize which are the 3 starts or the 3. It might be good to know that the start codon is this, but we don't need to memorize the 3 stop codons. And so this is a chart for helping us find which amino acid is coded by each codon. So let's do an example here. In this segment, let's just use the first codon, which is AUG, and let's find out which amino acid it codes for. So our first base would be our first letter, which is A. Second base, second letter, U. Third base, third letter is G. So then you follow them all back, you trace them back this way. This one, we have AUG right here, and it codes for a MET, which is our start codon. So a change in the order in which codons are read changes the resulting protein. So here we have reading frame 1 and reading frame 2. And scientists use reading frames to pair up or separate out each set of codons. So in this first frame, we're going to start with CGA, and that's our first codon, and then we keep going from there. So these are the proteins it codes for: ARG, TYR, SER, SER. So then reading frame 2, we skip the C because, it's a really long chain. It keeps continuing. You don't really know when it starts or ends. So we're going to start with GAU and then keep going from there. But our proteins change. Here we have ASP, THRVL. So it's very important that we start in the right spot. So regardless of the organism, codons code for the same amino acid. So this RNA could be in a rat, a chimp, a human. It'll all still code for ARG, TYR, SER, SER. Amino acids are linked to become a protein. An anticodon is a set of 3 nucleotides that is complementary to an mRNA codon. An anticodon is carried by a tRNA. So here we have tRNA, which is transfer RNA. It's just another type of RNA, and it helps in the translation process. So for the tRNA, it's everything but this amino acid up here. It's this part of the body and the anticodon. So tRNA carries an amino acid right here. It's carrying it, and it has an anticodon, which fits in with the opposing codons on a mRNA. So if mRNA has, let's say, AAA, the tRNA would be TTT, because A goes with T, C goes with G. So ribosomes consist of 2 subunits, and a ribosome is where translation happens. So we'll see a picture of it. The large subunit has 3 binding sites for tRNA, and the small subunit binds mRNA. So here's our green ribosome here. We have the small subunit where mRNA basically just runs through like a conveyor belt. And at the large subunit, we have binding sites for tRNA. So the tRNA comes and sits in these sites and matches up their anticodons with the regular codons of mRNA. So for translation to begin, tRNA binds to a start codon and signals the ribosomes to assemble. A complementary tRNA molecule binds to the exposed codon, brings its amino acid close to the first amino acid, and it forms a chain. So here's a really nice detailed picture. Everything starts in the nucleus, and then once we finished copying DNA and getting mRNA, the messenger RNA comes out in this long chain, which is basically the instructions from DNA. And it comes to the ribosomes, and it passes through the small subunit, and it just keeps passing through and passing through. And in the large subunits, we have tRNA, which come from the cytoplasm, and they come with their amino acids they have, and they bind to the subunits here, and they match up, they know where they match up, so they just keep going in like a factory, basically. They keep going, and they match up their anticodons with the regular codons, and once they match, they release their amino acid into this polypeptide chain here, and like the chain is really long, like this, and it forms a protein. So translation occurs in the cytoplasm of both eukaryotic and prokaryotic cells. It starts with a tRNA carrying a methylene attached to a start codon. So the ribosomes help form a polypeptide bond between the amino acids. So if we see here, all these little bonds between these amino acids are polypeptide bonds, which is very important vocab we need to learn. So the ribosome pulls the mRNA strand the length of one codon. So it's basically saying our ribosome here, it breaks the bond between the tRNA, the tRNA and the amino acid. It breaks that bond and it forms the polypeptide bond between the two amino acids. So the exposed codon in the first site attaches a complementary tRNA, brings an amino acid. The tRNA anticodon pairs with the mRNA codon, bringing it very close to the other tRNA molecule. So once one tRNA molecule starts, another one comes in right afterwards, and it brings its amino acid close to the other one, so the ribosome can form a bond between them. And as we see here, we can see how the anticodon and the codon works. So our codon is AUG, so our anticodon would have to, A would have to match with U, U would have to match with A, and G with C. So that's how they choose which ones go where. The ribosome forms a peptide bond between the two amino acids and breaks the bond between the first tRNA and its amino acid. The now empty tRNA molecule exits the ribosome. A complementary tRNA molecule binds to the next exposed codon. Once the stop codon is reached, the ribosome releases the protein and disassembles. So it's just showing how it's like a factory, and the tRNAs just keep coming, and they keep leaving. So only the middle site is used, they just keep switching in and out. And once the stop codon comes, so I guess this is, stop codon, UAG, once that stop codon comes, and the tRNA bonds with that, the polypeptide chain is complete, and the ribosome disassembles. So we're going to skip 8.6 because it's not important, and we're going to go to 8.7, which is mutations. Our key concept is mutations are changes in DNA that may or may not affect phenotypes. So as we know, the phenotype is how we look, and basically our body. And so mutations, as we know, we usually think of them as bad, but they're not always bad. They can be good. And a mutation is just a malfunction in our DNA. So some mutations affect a single gene, while others affect an entire chromosome. A mutation is a change in an organism's DNA. Many kinds of mutations can occur, especially during replication. So during the replication process, when we can copy our DNA, there can be some errors in that. A point mutation substitutes one nucleotide for another. So there are certain types of mutations we're going to learn about. First one is a point mutation, and this substitutes one nucleotide for another. So here we have our little diagram here. So as a mutation is a change in an organism's DNA, we already know that. So this is our normal DNA and RNA protein chart. So this is our regular DNA. We have G, A, T, C, T, and then we have a C after it. Here we have GA, which this is our point mutation, GATCT, and then we have the mutated base here. It comes in and it replaces the C that was here with the A. So this C is replaced with an A on the mutated part. And then our RNA changes because this A has to match with the U. And then our protein changes because the codon here is GAU, not GAG. So now we have ASP instead of GLU. So this protein change can cause a mutation, or it is a mutation, and it can either be bad or good. Many kinds of mutations can occur, especially during replication. A frameshift mutation inserts or deletes a nucleotide in the DNA sequence. So first we did a point mutation, which is it's not as lethal as a frameshift mutation, but it can still be bad. So a frameshift mutation inserts or deletes a nucleotide. So here we have the normal one, and here we have the frameshift mutation, and in this one it's a deletion. So here we had TCAGG, here we have TCAG. We deleted the first G here, deleted base here, and we just kept going. So we took out the G and the C here, and that causes our proteins to change. And so in some tests they might ask you what would happen if this is deleted or a point mutation occurred here, and you would just use the chart they give you for the proteins and find which protein it would match up with and see if, like they'll ask probably, did the phenotype of this species change, and if you turned out with a different protein it would, and if you didn't it wouldn't. So as you see here, with the deleted base we change more than one protein. In the point shift one, the point mutation, we only changed one amino acid and all the other ones stayed the same. But here in a frame shift, all the ones after the frame shift are going to change. Like, it's going to keep going because we changed our frame. Chromosomal mutations affect many genes because they happen in the chromosome. So they usually occur during crossing over. And gene duplication results from unequal crossing over. So here we have a picture of crossing over, but it looks a little different here because they don't look exactly the same but different colors. Here we had a very long X and now it turned short, and this one became even longer. So they crossed over, they had unequal crossing over, they didn't cross over in the right spot. So we have more genes on one and less on the other, so we have gene duplication here. Translocation results from the exchange of DNA segments between non-homologous chromosomes. So these are our sets of chromosomes here. 2 long ones, 2 short ones. So the 2 long ones would be homologous and the 2 short ones would. And so ideally these 2 would cross over and these 2 would cross over. But that's not what happens in translocation. Translocation, this is our translocated one. The long green one did crossing over with the small red one, which shouldn't happen. So this is our turnout, and that's really bad because it affects so many genes from the chromosome. Mutations may or may not affect phenotype, as we talked about earlier. Chromosomal mutations tend to have a big effect. So, as we say it may not affect phenotype, let's go back to the point mutation. So, this point mutation here, that's frame shift. Point mutations, it changes one thing, but sometimes it could change it, and the ending protein could still be the same because there are different codons that code for the same protein. So sometimes this will change, but we'll have the same protein end up. And there's usually questions about that too. So you gotta know that it's not always gonna change. I mean, you can have a mutation, but you just can't see it. So some gene mutations change phenotype. A mutation causes a premature stop codon. A mutation may change protein shape or the active site. A mutation may change gene regulation. So there are many bad cases of mutations, but there could be some good ones. And this is just an example. Blockage, no blockage. Some gene mutations do not affect phenotype. A mutation may be silent, so if it's silent, that means it doesn't affect. and it may occur in a non-coding region. A mutation may not affect protein foldings or the active site. So protein foldings was when we're making the amino acids, when I talked about the point mutation, that you can have a mutation but you'll still end up with the same protein. So mutations in body cells do not affect offspring. So if it's in your body cells, so everything but your sex cells, you won't pass it on. But if it occurs in a sex cell, it may be harmful or beneficial to offspring. Natural selection usually removes the the harming mutations, because if you already learned about natural selection, it's like survival of the fittest, and if the mutation isn't good for you, you're probably going to either die off early before you can reproduce, or nobody's going to reproduce with you. So they can be caused by several factors. Replication errors can cause mutations. So when our DNA is replicating, some error goes on in that, and you like mistake a A for a G or something, and something bad happens. Mutagens such as UV rays and chemicals like cigarettes or abundance of bad alcohol and stuff like that. So some cancer drugs use mutagens to properties to kill cancer cells. So sometimes scientists can use these to reverse bad. And so that's the end of chapter 8, which was all about going from DNA to proteins. And next we will be doing chapter 9, so stay curious and watch the next video.